All right, and we'll start just by going over the warm-up again. Let's do that. Share screen. And quiz statistics. And as I say each time, and I'll say one more time, like if you can take a look, we're getting this kind of bimodal thing again. Some people are getting like less than half. Fortunately, most people are, getting most of them. If you're down in this area, again, the next big exam is 1 week from today. You really got to make sure that you're getting up to speed on things or it's not going to go well. Because we're going to be covering even more material today and on Thursday. That will be on your exam on this coming Wednesday, Tuesday. All right. So receptors responsible for the general senses. Here, most people got this. Typically very simple structures. They talked about a lot of them are literally just naked dendritic endings. And even the complicated ones are little dendrites with a little bit of connective tissue capsules on them. And they're all over their body. They're not just in the head, and they all respond to different kinds of stimuli. There's thermoreceptors and touch, deep pressure, light touch, pain receptors, a variety of stimuli they respond to. And again, not autonomic. Autonomic division, I mentioned back at the very beginning, is motor. So anything that has to do with sensing is not going to be autonomic division, because autonomic division is about activating things. So you want to keep that straight. Choose all the senses that are mediated by mechanoreceptors. So this is good. Most people got this. Hearing, which we're going to look at in much more detail today, is sensing vibration. So it's actually mediated by little receptors that detect bending, that ultimately is being activated by the air pressures moving your eardrum. And touch, obviously. This one, yeah, most people got this as well. Aqueous humor being made and drained, produced by the ciliary bodies, drained by that scleral sinus, that canal Schlemm. What is the pathological condition if the drainage is blocked and the pressure starts building up?
Glaucoma.
Glaucoma. This one, definitely no capillaries within the lens and cornea, because that would be like that would kind of make it hard. Those are the clear structures that the light has to get through to get to your eye. Gustatory receptors have the ability to recognize. So this one, yeah, most people got this correct as well. We talked about taste receptors only have like 5 core tastes that they discern, whereas the smell receptors, the olfactory receptors, have a much wider range of different molecules, and that we sense as, roses or violets or skunk or urine or decaying flesh or whatever. But there's a much broader range of molecules that we can discern with olfaction. And this one, yeah, most people got this as well. Olfaction is kind of the special sense in that it projects into the brain in a lot of ways, like getting into the limbic system, all these kind of subcortical going into parts of the brain without first going into the thalamus, which most senses do. So that is that. Well, also reminding people that this introduction to research paper and presentation, choosing a topic, that is due this week. So I think I have it due on the 29th, so a couple of days from now. Some people have already turned these in. But if you have not, make sure you get on that. My plan is on Thursday to go through them and give everybody feedback on them. And then once you've got an approved project, you'll be able to kind of move forward and start working on that. But just reminding people that that needs to happen.
Dr. Egger, if we have like a couple things in mind, can we write a couple like things down or a few things down and then you pick the best topic?
Yeah, absolutely. Absolutely.
Okay.
Yeah.
Okay, thanks.
Or I might say they all sound good, or I might say this one sounds better than this one. So either way. Yeah. And remember, it's a proposal. So it's like just like a sentence or two. This is not like a big developed thing. You're just wanting to go out there, double checking that there actually is peer-reviewed research that is going to allow you to do this project properly. And we'll take it from there. What else? Yeah, I mean, a few people were emailing me about the lab today. Today's lab does actually have a objective and hypothesis and all that, but we're going to do it together when we get to lab. Today's lab is going to be like an exercise in kind of getting more down and dirty with how do you design an experiment? Because up to this point, all of our labs have been kind of more cookbook, these recipes. They're kind of, you open up your lab manual and it tells you what to do. And obviously in real science, you actually have to figure out like, how do I actually test my hypothesis? How do I design my experiment? So that's something we're going to work on together in lab. So that's why there is no entry in your lab manual for today's lab, because we're gonna work and develop it all together. So some people, said, is there anything I got to do? And I said, no, in case you were like in the back of your mind worrying, did I miss out? Did I miss something I was supposed to do for lab? You weren't. All right, so it's time to continue. We're going to finish off let me stop sharing this. We're going to finish off the nervous system finally. We still got to finish the eyes, do the ears, and then we'll be able to get into the muscles. And before this week is done, we'll have finished the nervous system, which we have done in quite a bit of depth. And we're going to do the muscles. Those are what's going to be on exam 2, which is on Tuesday. So Tuesday is going to cover everything from the nervous system that we've been covering since the first exam, as well as the muscles, which we're going to be covering at the end of today and then on Thursday. So let's dig in. So we're just going to continue on the special senses. Again, we did what were the 2 special senses that specialized in chemoreception?
Olfaction, gustation.
Yeah, that was olfaction and gustation. And how do these 2 senses actually accomplish their sensory transduction?
Olfaction by cells that are in the roof of the nose. Are specialized, and thus they do the sensory transduction.
And what exactly are those cells doing? What's happening at those cells to actually allow the signal to be transduced?
Chemical binds the receptor.
There's a receptor. There's receptors on the membrane of either these olfactory cells or these gustatory cells, depending which one you're looking at. And when the molecule, the appropriate ligand, binds that receptor, that's going to start some pathway. It might start a second messenger pathway that opens up ion channels, or maybe some of them actually directly result in the release of neurotransmitter from the cell, which is then going to activate action potentials in the subsequent neurons. But either way, in both cases here, what is the stimulus? The stimulus for these senses is
Molecule.
Molecules, chemical molecules.
kinds of molecules. You know, the transduction happens by having receptors that recognize and bind those molecules, and then, you know, ultimately turn on some pathway that results in some electrical signal, some action potential, zipping down some sensory neuron going towards the CNS. So that's that. We're going to be doing today is going into the eye and into the ear and looking at how the eye changes light into some signal which goes into the nervous system. You know, so far we've been talking about how the eye kind of focuses the image, how it adjusts the image, but we haven't actually talked about how the eye converts light into electrical signals into the nervous system. So that's what we're gonna dig into now. And then once we're done with that, we'll get into the ear and look at how the ear manages to convert kind of the movement of your head or the pull of gravity or the vibration of the air into electrical signals going into your brain. So now it's time to go continue on the eye. You know, there's lots of stuff we can talk about the eye that we're not in the interest of time. So we have talked about the fluids in the eyeball, that aqueous humor being produced and drained, the vitreous humor, which is more kind of that jelly-like stuff kind of supporting the inside. When we get to the immune system later, we're going to talk about things that keep you safe from pathogens. I mean, one thing though, you have like a lacrimal gland, you actually have this lacrimal solution, your tears constantly being produced and flooding over your eye, and they're also being constantly produced and drained. You know, the lacrimal gland here. You have these little lacrimal puncta, if you remember from anatomy, that just drain into your nasal cavity. So tear solution is always being produced, kind of irrigating, keeping your eye moist. That lacrimal solution has lysozyme, actually it's antimicrobial stuff, which helps keep your eye safe from, you know, little microbes that might land in your eye or something. What we're going to do now, though, is there can be like somebody diving down through the pupil. And the pupil is just a hole through the iris. And now we're inside the eyeball. There's our little guy diving down. Woo. Here was the iris. And on the back of the eyeball, the innermost layer is the retina. Again, we have the lens here. So the lights, we've talked a bunch about how the light comes in, it gets focused. What we're going to focus on now for the rest of our discussion here is what's going on at the retina. This is where the actual phototransduction happens. phototransduction, the specialized version of sensory transduction for light. There are a variety of different cells. If I take a little chunk of the retina and kind of blow it up, so retina detail. So this is the front of eye where the light's coming in from. The retina has rods and cones. These are the actual cells that do the phototransduction. These are the ones we're going to look at in even more detail, because these are the ones that literally convert light into signals that start percolating through the nervous system. The retina also has lots of other cells in it that we're not going to go into too much detail. There's also bipolar cells. There's the ganglion cells that actually make the optic nerve. There's also these things called Amacrine cells. I'm not going to get into this. There's little horizontal cells. The reason why I am kind of mentioning this whole zoo of cells, though, is just to give you this idea that the retina is doing more than purely phototransduction. During development, the retina develops as an outpocketing from the diencephalon, right? The same part of the brain that becomes like the hypothalamus and thalamus and everything actually kind of migrates forward a little piece of that. People sometimes talk about like the retina, an approachable part of the brain, because it's actually migrated out into behind your eyeballs. So some of the very fundamental like kind of signal processing actually happens in the retina itself, kind of sharpening boundaries, enhancing contrasts and things like that. So I'm, again, in the interest of actually making it through everything we need to get through, I'm not going to go into a lot of detail there, but these cells are actually doing some useful stuff to kind of tighten up the signal, enhance boundaries and things like that. So the retina, as opposed to some of these other receptors that we've been talking about that are just about convert the stimulus into a signal and send it to the brain, the retina actually has a little more going on, actually a lot more going on. Like there's miles, I think there's literally miles of like dendrites and stuff if you add up all of the wiring inside a retina. And you've got something like 250 million photoreceptors in your eyes that are doing this phototransduction. One of the weird things in the mammalian eye is the fact that the light is coming in and all of these cells are actually in front of the rods and cones that actually do the phototransduction, which means the light actually has to come through these other cells to get to where you convert it. Fortunately, the cells are, clear, so they don't really block much. And also in the middle of the retina, the place we're going to see the Fovea Centralis, where you have the most acute vision, all of these cells tend to get kind of pushed to the side to try to make it as thin of a layer of distortion as possible. We'll talk a little bit more about the Fovea Centralis in a few moments, but it's kind of a weird thing. It's also why you have the blind spot, since the ganglion cells that make the optic nerve are actually in front of the photodetectors. Then the axons have to go out the back to get to the brain, which means there has to be some area with no photodetectors. again, there's so many cool things about cephalopods, about like octopi and stuff, but one of the really cool things about an octopus or a cuttlefish or any of those dudes is they have really cool eyes, but the rods and cones are in the front. So they don't have a blind spot, right? Because they just have all the wiring going out the back. You know, I have no idea. This obviously works for us, but it's kind of a weird design. So rods and cones. Rods and cones, I'm going to first talk about them in a more general way, kind of what they do, how they're laid out in the retina. And then we're going to get into kind of real detail about what exactly has happened when light energy hits one of these cells and what is the actual pathway that actually creates the signal that is going to become the phototransduction. But we're going to start a little kind of step back a little bit. So rods and cones. And they're also going to be obviously at the core of our lab today. Our lab today is going to be playing around with properties that distinguish rods and cones. So how are they both similar? How are they the same?
They're photoreceptors, both?
Yeah, they're photoreceptors, which means they both detect light and can convert that into some signal that it sends into the nervous system. So that's the same thing in both of them. And we'll see actually the basic mechanics are the same in both of them, although some of the details are a little different. So they're both photoreceptors. So they both detect light and create a signal. But then there's some pretty major differences. Rods are active in dim light. Cones are active in bright light. So it's like sometimes when on like some exam where I say, give me different, compare and contrast rods and cones, and people like, rods work in the dark and cones work in the light. You know, that's not true. If it's totally dark, nothing's gonna work, right? Dark, absence of light. The rods work in very dim light. Like they don't need much light to actually detect that there's something there. But they need light. They don't work without light. That being said, they've done experiments where they can see a rod actually having a measurable response to a single photon of light hitting it. You know, it's going to be because there's a second messenger pathway with amplification, which, you know, so that shouldn't sound all that surprising that one tiny little event can actually have a measurable effect if we're using second messenger pathways. But when we get to the actual phototransduction, it's going to be a G protein mediated pathway. So bright light, dim light. You know, right now, if you're just kind of watching me on the screen, you're using your cones. But like when you're running around in the moonlight and everything is kind of dim outside, then you're using your rods. The cones don't work in dim light. Cones, color, vision. I've talked about this in lab. There's 3 different versions. There's the red cones, the green cones, the blue cones. You know, depending on the frequency, you know, what we think of as color of light is just the frequency. Kind of the same way in the hearing lab, we talked about pitch. When sound waves are higher or lower, it's like, it's like high pitch, low pitch. When electromagnetic waves are higher or lower frequency, we see them as more purple or more red. is just the way our nervous system creates a subjective experience of the frequency of the electromagnetic radiation coming in that's detected by our eyes. You know, what does it mean? Like I can look, I'm looking at the screen. You know, I can see, like Emily has kind of a greenish sweater on. You know, why does her sweater look kind of greenish to me?
Because your cones are, or the green cones are being stimulated by that frequency of.
Yeah, so basically her, the pigment, the dye in her sweater is absorbing those other colors and reflecting back the green light into my eyes, which makes my green cones get excited and I see green. It's the same way plants look green because the plants are actually using the red light for photosynthesis. So they absorb the red, but they're throwing away the green, which is what ends up on your eyeballs and activating your cones. Like Mason's wearing a black hoodie. Why does his hoodie look black?
In all the colors.
What's that?
It's absorbing all the colors.
It's absorbing all the light, so nothing's coming back. So none of my cones are getting excited. Whereas I see Wendy's got a white sweater on. What does white mean?
No colors absorbed.
Say it again.
There isn't any colors that are being absorbed.
Yeah, so that means all the colors are coming. And again, it's a little non-intuitive because when you're mixing light or mixing luminance, it's different than when you're mixing pigment or paint, right? If you mix all the colors together, if it's paint, you just get muck. But if you mix all the colors of light together, you get white light, right? You've seen this. If you like shine sunlight through a prism, it breaks apart into all the different colors. So it's, you know, you do have some intuition that white light, sunlight actually has all the colors all mixed up into it. So rods, on the other hand, there's only one kind of rod. They absorb primarily in kind of the greenish light, but I'm just going to say they're monochrome. They can tell that there is the presence of light or the absence of light, but they can't tell you more subtlety about what frequency is it. Is it high frequency, low frequency? So they're monochrome. And again, they absorb more around in the green, which is part of the reason why you have like safe lights. Like when you have like emergency lights, they're usually red because the red is actually not gonna bleach out your rods because the rods don't even know red exists. So rods and cones, color vision, monochrome. Another thing is cones are high acuity. You know, the same way we talked about like with touch discrimination, can you tell if those two needles are really close on your skin? Is that one needle or two needles? High acuity, are those, is that two dots or one dot? Can you determine like very fine detail in the visual world? Rods are low acuity. You know, there's something there, but I can't really tell you much more other than there's something there. We're going to talk a little more about how that actually happens. But in general, rods will tell you, you know, there's, you're not going to walk in, like break your nose on the tree. You're not going to walk into the tree, but you're probably not going to be able to tell the pattern on the bark. In terms of how they're actually laid out in the retina, the cones are found in the center of the retina. The rods are in the periphery of the retina. So if I go back to the picture of the eyeball, here's my retina. The cones are going to be, and actually I made it, it's actually too big. It's actually just even a smaller, it's a tiny little dot, little zone where the cones are. And then the rods are going to be scattered all around more on the periphery.
So that center of the retina, is that a place where the macula lutea is?
Exactly.
Okay.
So the macula lutea is the center, and the center of the macula lutea, which is something you should know, is the Fovea Centralis. Fovea, it means like this spot in the center. And it's the most acute vision here. Basically pure cones. So when you stare right at something, the image lands on the fovea. It lands on this area that has this dense pack of cones, which are used for very acute vision, for fine detail. In fact, in vision science, the verb we use, if we want someone to stare right at something, we say we tell them they're foveating. To foveate means stare right at it, so the image lands on your fovea centralis, which is where you're going to see the most detail. The rods on the periphery have less. Again, if you just put your hands, if you stare straight at your screen, but notice your hands off to your side, like put your hands to the side of your face and wiggle your fingers around, you'll know there's hands there, but you can't even really notice their fingers on there. You can tell there's something there, right? Right. So that's this idea of rod vision. You can tell there's something there, but you can't really tell a lot about the detail of it. Normally, if we were in school, I would like shut the blinds and turn off the lights, and we would actually go back and forth between rod and cone vision. Instead, I'm going to give you a little exercise you can do at home to kind of play around with it, because it really is worth kind of noticing how different the two types of vision are. Like right now, when it's bright, your rods are pretty much bleached. They're doing some basic transduction, but for the most part, they are overwhelmed by this bright light. But your cones are totally rocking it. And so you're like in cone vision, things are sharp, things are color. But if you turn off the lights, then the cones are useless. And the rods have been what we call bleached. Most of the photopigments have been we're going to talk about it. They've been kind of transformed. They got to take time to recover. It takes maybe up to like 20 minutes. Within 5 minutes, the rods are actually working pretty well once the light becomes dim. But they keep getting better and better. We say like dark adaptation. So in bright light, cones are active, but the rods, we say they're bleached. The pigment gets transformed into this version that needs to get reactivated. And in fact, it's literally bleached too. Like when we talk about the visual pigments, they used to call it visual purple, because when they would cut open the eye of some little animal, it would be purple, but then as soon as it was exposed to light, it would like lose the color, it would bleach out. So there's literally a bleaching that happens. So rods, dim light, do not have much detail. And part of the reason they have a lot of sensitivity, but they don't have a lot of like visual acuity, is there's a lot of convergence in the signal pathway. Like if you might have hundreds of rods that all come into the same axon, which then goes to the brain. So there's a huge array of receptors that are out there trying to like notice, is there a photon landing in the retina? So you have a much better chance of noticing light, but you don't know which of these, where did it come from? Like exactly the localization is not clear. Whereas in the cones, basically each cone gets its own personal axon to the brain. So you have, was it that cone or that cone or that cone? You have very good like discrimination in terms of the acuity, but it means you need a lot of light, a major signal coming in to actually activate that particular cone. So part of the reason rods are more sensitive than cones is just in the way they're wired like this.
Wait, sorry, Professor, you just said that rods have more sensitivity to cones. Do you mean cones have more?
Rods are much more sensitive to light.
Oh, to what? Sorry. Okay, thank you.
Yeah. Cones are much better in terms of visual acuity and in discriminating fine detail.
Got it. No, I just got confused.
And so this, like, if you're like if people do stargazing or astronomy, like what's the little trick? If you're trying to see like a very dim star in the sky, do you look straight at it?
No, you keep it on just outside your center vision.
Yeah, you always look to the side of it. Why would that make sense that you don't actually want to look straight at a dim star if you actually want to see it?
So the image doesn't go to the cones, just perceived by the rods?
Exactly. If you have some really dim star here and you stare right at it, it's going to land on the cones and it's not going to activate the cones. You're not going to notice it. Whereas if you look off to the side, and then this is actually coming and landing on the periphery, on the rods, then it actually will activate your retina. And if you've never done it, it's really weird. You can like stare at this thing, it disappears, you look off to the side, it shows up again, and you stare at it, it disappears, you look off to the side, it shows up again. So that is kind of the story of rods and cones. We're gonna be thinking about these different properties when we're designing our lab later this morning. So the last thing we want to do now is go into the detail of what is happening in a rod or a cone in order to actually do this phototransduction. And it's going to be nice. It's going to bring back your old pals, second messenger signaling and G proteins and phosphodiesterase and cyclic GMP and all that stuff. So let's look at this. So the basic story is the same, whether you're looking at a rod or a cone. There are a few little different versions of the molecules involved. The wiring's a little different for rods and cones, but the basic story is the same. So I'll draw a rod. They're weird cells. This is like a rod. They're called rods because this part they call the outer segment looks kind of more rod shaped. In cones, it's got a little more of a pointy head, so it's called a cone. It's got lots of folds in it to increase surface area, so we can have lots of these membrane proteins that are going to actually being there to detect the light. And now we kind of got to lay things out here. So I'm going to have little sodium channels. So in the dark, let's start in the dark. In the dark, we have our cell here. There's cyclic GMP inside the cell. You remember cyclic GMP is just one of these second messengers. That cyclic GMP is responsible for keeping these sodium channels open. So we get what's called the dark current, because you have sodium channels open. What happens to the membrane potential if sodium is coming into a cell?
Increases.
It increases. So this is actually going to be a little counterintuitive. If, usually you think about when something gets excited, the voltage goes up. It turns out in the eye, it's going to be the opposite. When this thing is not excited, when it's just in the dark, we have what's called this dark current. Let me see if I got the actual voltage here. Yeah, it's around. So VM goes up to around minus 40 millivolts. So in the dark, the voltage is actually more positive than resting, right? A normal resting cell is around minus 70. So in the dark, we actually have it's depolarized. This thing is actually releasing glutamate. Which then is talking to the bipolar cells and the rest of there's actually no action potentials in the retina until you get to the axons of the ganglion cells. But so this is, again, a little counterintuitive. When we're in the dark, the cell is actually depolarized, which is causing it to release its neurotransmitter. But because the retina is a complicated neural network, it knows that that means dark. It knows that when the voltage is higher on the rod, that we're in the dark. So now we need to talk about how does light fit into this. So the light is going to be detected by membrane proteins up in this upper part, this outer segment. So there are going to be these proteins called opsins with this other thing called retinal on them. So again, this is in the membrane. Opsin is a membrane protein. And opsin is kind of like an antenna that absorbs light in different frequencies. When we get to different cones that absorb different colors, it's going to be because they have slightly different kinds of opsins that will get excited, absorb the light energy at different frequencies. The retinal has two conformations. Remember, conformation, you can have molecules that can flip, and you can have the same molecule, but it can bend in one way or bend in another way. So retinal has what's called a cis conformation, cisretinal. This is kind of when it's ready to go. If it absorbs light, it actually snaps into this trans conformation. And this is going to be at the core of this whole process of phototransduction. We basically, in the membrane of the rod, we have this little thing, again, this opsin and retinal unit here. And again, it's kind of acting like a little antenna, absorbing the light energy. When it absorbs the light energy, cisretinal turns into transretinal, and that shape change is going to now start a second messenger pathway. So transretinal is going to be, so right in most of the signal transduction we've been seeing, there's some ligand that binds a receptor, and then that's going to trigger some second messenger pathway, because ligand binding the receptor changes the shape of the receptor and activates the G protein, and you remember all that stuff. In this case, instead of having a receptor that binds a ligand, we have this opsin and retinal complex here that absorbs light energy, causes a conformational change in the retinal, and that's going to trigger our second messenger pathway. So this shape change is going to cause, in fact, maybe I just, so if this is 1, let me give myself a smaller, so if this is 1, cis turns to transretinal. 2 would be, retinal detaches from opsin. And now we basically have, this is going to, this activated opsin is going to turn on a G protein. And the G protein gets a special name called transducin. And so this is now activating. This is just a second messenger pathway here. Right, so we don't have to go in fact, I'm not going to go into the details, but when you turn on a G protein, you then start a whole cascade of events. That G protein activated cascade is going to ultimately activate phosphodiesterase in the cell. What does phosphodiesterase do? We've met phosphodiesterase before.
Stops
Breakdown
UGM.
cyclic GMP.
It breaks down cyclic GMP, right? So 6, cyclic GMP is going to get broken down. And once cyclic GMP is broken down, what's going to happen?
The stops.
Transduction stops.
Well, the sodium channels are going to close. No, transduction, no, this is transduction. Transduction doesn't stop. This is transduction, like up close and personal, happening right now. So the sodium channels, which were held open by the cyclic GMP, are going to close. And when the cyclic GMP is broken down and the sodium channels close, what's going to happen next?
VM goes down.
So that's going to be 8. And that is indeed the voltage change that is the phototransduction.
I'm sorry, Dr. E, what did you write for 8? Because I can't see it. Can you repeat that?
VM is going down.
Okay, thank you.
So the membrane voltage. So remember, sensory transduction is the voltage change. Convert the signal into some electrical, convert the stimulus into an electrical signal. So we have done it. It's upside down in terms of the voltage. The voltage goes down in response to light, but it's still, the voltage is modulated in response to the stimulus. So, and you should know these steps. Like I could easily on an exam, say on Tuesday, ask you to explain how phototransduction works, right? So, all the stuff about the lens and everything, that's how we get the light focused. But in terms of actually creating an electrical signal, that's this mess. If you want to understand phototransduction, how do we actually convert light into an electrical signal? You've got to realize that it starts with, kind of light coming in and light getting absorbed by the opsin, and then retinal getting transformed into cisretinal, from cis into transretinal. So then we have this activated opsin, then activating a G protein, and the G protein turning on a second messenger pathway, which turns on phosphodiesterase, and then phosphodiesterase going and breaking down the cyclic GMP. And without cyclic GMP, the sodium channels close. When the sodium channels close, the voltage of the cell goes down, and that's the voltage change that is the transduction. So it's, there's a lot to it, but it should feel a little less like overwhelming now that you actually are kind of familiar with this idea of these crazy second messenger pathways and things going on, these cascades and metabolic pathways and all this stuff. So now this is just another flavor of something that you've already got some familiarity with.
Professor, I have a question. Could you just go over, I'm a little confused just in the beginning part when you were talking about in the dark versus not.
Mm-hmm.
Your first part, and that you were like, it's a little confusing.
So in the dark, sodium is coming in. VM is like minus 40, which is kind of positive. Due to influx of sodium. When light hits this thing, it's going to do that whole thing. Retinal cis to trans, activated opsin turns on the G protein, turns on phosphodiesterase, breaks down the cyclic GMP, closes the sodium channels. And now, in response to the light, VM drops down to minus 70 millivolts. And that voltage change from this more positive to this more negative is the signal transduction.
Okay, that makes sense. So I think what I was getting confused about was before the influx of Na+, it was a lot more negative. And then the influx of Na+ made it more.
Correct.
Okay.
Like if you shut those sodium channels, the voltage drops back down to like minus 70.
Right. Okay. Got it. Thank you.
Sure. And like I said, it's a little counterintuitive because you like to think about, oh, we excited, it's going to get more positive, but it's the opposite. You excite it, it actually gets more negative. But the system is designed to roll with that. It knows what it means. So you detect that as the presence of light in your vision.
So one question. So before even the sodium channel comes in, opens up, the glutamate, the neurotransmitter, is released for excitation?
Yeah, so when this happens, you actually have less glutamate being released. And that's the bipolar cells know that that is the sign that the light has hit. There's basically, in the circuitry, there's an inverter, things that turn absence into a positive signal, right? And if you do circuits and stuff, you have inverters all the time, things that turn ones into zeros and zeros into ones. That's just a typical part in signal processing and stuff like that. So it basically inverts the signal. So it, negative means yes, positive means no. It's really easy to make a little circuit that can invert signals like that.
So less glutamate, and then sodium channels opens, and then all the thing happens, right?
Wait, say it again.
So when the phototransduction happens, the glutamate is lessened, the release of it. Okay.
Yeah. So the sodium channels close and there's less glutamate being released as well. All right. Very briefly, I'll just, I've mentioned this before in lab, but it's kind of worth mentioning here as well. It's the idea of how the eyes feed into the brain. Like if these are your right and left hemispheres, remember the occipital lobe, you have the primary visual cortex, your retina on the back of your eyeballs. The thing that is different about how the eyes connect to the brain compared to most other things is, the right and left, the lateralization still happens where you have things represented on the opposite sides, but it's the visual field. So things that are happening on this side of the world, which is coming onto like this side of the retina, is gonna both eyes send the message to the same side of the brain. Things that are coming in from the other side of the world, which is coming onto the other side of the retina, those are all going on to the other side of the, right? So that's how that kind of binocular stereoscopic vision works, is each hemisphere is actually getting a slightly different view of the same thing from that same side of the world. And again, going through the thalamus. In this case, there's these nuclei. The lateral geniculate nucleus is the specific part of the thalamus that it's going through. So vision. So let's move into hearing. Are there any last questions about the eye before we roll along? Okay.
Oh, I have a quick question. So no, we're not going over cones or anything, or do they act similarly, or?
The cones act similarly. They have a different version of opsin that absorbs light in different frequencies. And then, like I said, they're wired differently, and the way they're just wired in the retina and the convergence of the rods versus like single axons for cone, or that are kind of the downstream from cones, makes a big difference in how they respond as well.
Okay, and then if for the stimulus for a rod, are you expecting like photon, light? I guess like what is the exact stimulus that we're kind of looking at? Just so I know to like, it's like a list stuff.
Right. So if you really want to get into it, you know, it's electromagnetic radiation. It's kind of undulating fields of electricity and magnetism going through space. You know, what exactly that is weird. If you've taken much physics and get into, are there photons, are they waves, or what, it's all weird. You know, it's,
So like what would be like a multiple choice option and like the right answer on your test if you were to write one about this? Like, would you say light, photon?
I mean, all of those would work, right?
Okay. Yeah.
I mean, I'm not trying to fool you there. You know, electromagnetic radiation is more than just visible light, though. It includes like X-rays and it includes radio waves. Like
Mhm.
the difference, if you look at the in fact, here I'll share real briefly. This is something I have for you to look at later, but I can just show it to you now because it'll be useful. This idea of the electromagnetic spectrum, it's basically these kind of undulating electromagnetic fields going through space, and depending on how fast they vibrate, you know, if it's in this narrow range of frequencies, we detect it with our eyes and we call it visible light. If it's a little lower frequency, we feel it actually. It activates our temperature receptors. We feel it as heat. When it gets much lower, it becomes microwaves or radio waves, which we can't detect, but you know, microwaves actually will get absorbed by water molecules and start heating them up as they absorb the energy, which is why microwave ovens work. If you start speeding up, go at higher frequencies, you get to ultraviolet. You know, some animals, like, you know, bees and stuff, their eyes actually do detect higher frequencies. They look at flowers that have patterns that make them look like cool bullseyes if you actually can see in the ultraviolet. For us, they just look white, but the bee sees this cool bullseye. As you get further up into X-rays, gamma rays, you're getting higher and higher frequency. These get pretty dangerous because they're got a lot of energy and like rip your rip your atoms apart and stuff.
Okay, thank you.
Yeah, so, but visible, so if you want to, get into the physics of it, it's this weird stuff that's electromagnetic radiation, and the majority of the electromagnetic radiation that lands on the earth generated by the sun is in this very narrow frequency range. So I think that's why we evolved to have receptors that are particularly nuanced at detecting that range of it. But it's just, visible light is no different from radio waves or X-rays than like D is from do is from da, you know, in terms of, you know, sound waves. They're just different frequencies of vibration.
Mm-hmm. Okay, thank you.
But that being said, even if you take advanced physics classes, it's just freaking weird stuff. Let's see. So we are now going to get into hearing and equilibrium. You know, like you put them together because these are both done by your ear. You know, your ear obviously does a special sense of hearing, of sound, but that's like one part of the ear. That's the cochlea. The cochlea converts, you know, vibrations in the air to the experience of sound. But the ear also has the saccule and the utricle, which detect the pull of gravity. It has the 3 semicircular canals, which detect rotational acceleration of your head. So we're going to look at all of that. At the core of it all, it is still mechanotransduction, and it's all based on this cool little cell called the hair cell, which is a mechanotransducer, something that converts mechanical stimulus into an electrical stimulus. So we're going to meet the hair cell. It's called a hair cell because it has these are actually stereocilia. This is a true cilium. They call this the kinocilium. It's the tallest of the hair bundle. And then the rest of the hairs kind of come down as a kind of in a tiered kind of structure like this. This is usually paired up with some neuron here, which is axon that's going to take the signal away. And the basic idea here is this is a mechanoreceptor. When you bend the hair bundle towards the side with the kinocilium, this is going to depolarize the hair cell. When you bend it away from the kinocilium, you're going to hyperpolarize. Depending on whether it's depolarizing or hyperpolarizing, that's going to change the probability of action potentials getting sent down to the brain. So the brain knows which way is this thing bending. Is it bending this way or is it bending that way? Or is it not bending at all? So this is going to be at the core of whether you're talking about hearing or any of these balance senses. Again, I could talk forever about these. I literally spent like 3 or 4 years of my life. So my whole dissertation work was looking at details of hair cells and how they do what they do and all this. But I'm going to fight the urge to talk about them more and move on here. I will say though, aren't they pretty? We're gonna go on. Now these kind of remind me of Bart Simpson, kind of a little Bart Simpson hairdo or something. So the ear at the core is gonna be the hair cells, but we also should talk about the other just like with the eyeball, you know, the core of signal transduction in the eye is the retina, but you got to talk about the iris and the lens. For the ear, the core of hearing is in the hair cells in the inner ear, but we should talk a little bit about the outer and middle ear leading in. And again, we've talked about this quite a bit in the lab. So rather than starting from scratch, I'm going to just bring up a, I have like the PowerPoint things from the class, from the book. So we can, let me share this. The ear. So this should look kind of familiar because you've already seen this in lab and you've got already some basic idea of how this works. Since you had to understand the difference between, sensory neural, remember the inner ear, this is the part where you actually have the sensory transduction. If there's damage in here, you had sensory neural deafness. Whereas this middle ear, that's where you had conduction deafness. If there was some problem, if the signals coming in, the vibrating air, it's going to vibrate the tympanic membrane, the eardrum. That then gets transferred through these little ossicles, the malleus, the incus, the stapes. In the common tongue, it's the hammer, the anvil, and the stirrup. They're basically like little levers. Like the hair cells, this is where you find the hair cells. The hair cells are in here. So the place where actual sensory transduction is happening is down here. In the inner ear is fluid filled. We're going to talk about the endolymph, the perilymph. The hair cells are all bathed in fluid. The little hair bundles are waving back and forth in fluid. However, obviously, sound waves are coming through the air. So a big part of what this middle ear is doing, in engineering, we call it impedance matching. We're making sure that we're efficiently transferring the energy that is the air vibrations and creating these vibrations in the fluid environment within the cochlea and the inner ear. Like if you just try to have the air vibration like hit and just directly go in, a lot of the energy would just bounce away. It wouldn't actually efficiently go in and create, you know, waves within the fluid within there. It would just kind of bounce off. So the little system of ear bones lets you efficiently transfer the air vibration into the fluid filled world of the inner ear. There's this little, what's called the oval window here. The oval window is where, maybe I changed my color here. Oops. Oh, come on. Oh, there it is. Make it green. Kind of right in here is this oval window. That is where you actually have a little membrane where if you push and pull there, you can send these vibrations into the inner ear. Because fluid is not compressible, you can't push if there's nothing to give. So there has to be another little window, this little, oh, in fact, there's the name of it, the round window. The round window is the thing that gives. If you push in on the oval window, oh here, oval window. If you push in on the oval window, then the round window can pooch out a bit. You know, if you pull in on the oval window, the round window can suck in a little bit. You know, so it's made so you can actually create these waves in this fluid thing by pushing and pulling on the oval window here. So this is going to be important when we get to hearing. And again, there's other things going on in the inner ear here. The balance senses don't care about the vibrations, but for right now, this is, if you're talking about the hearing part of the ear, you need to think about transferring the vibrations from the air into the inner ear. The other things I'll talk about just briefly before we leave the middle ear. Like outer ear, you know, we don't have to talk too much about, but I will mention if you're a non-human mammal, like if you have cats, you probably notice cats have a lot more control over their pinnae, right? Cats, you have their ears totally move towards you and kind of funnel in the sound. You know, our ears, people who practice can wiggle their ears a little bit, but in general, humans don't do such a good job of that. But that doesn't mean that lots of other animals don't use their pinna. Cats use their pinnas for nonverbal communication all the time, right? The airplane ears, I'm pissed at you, or go up like I'm like paying attention to you or whatever. So pinnae are important, but not so much for as much in humans. Middle ear, transferring the vibration efficiently into the inner ear. There are a couple of little muscles that can actually tighten up. One of them, if I'm not normally I would type it up. Maybe let me go back over to here. There's one little, whoops. There's one little muscle called stapedius. Another one called tensor tympanum. These are little muscles that can tighten up either the tympanic membrane so it doesn't vibrate as much, or tighten up the little stapes so it can't transfer the vibration to the inner ear. This is part of the way you have to protect from damaging the more delicate hair cells in the inner ear from really loud sounds. Basically, if you tighten these up, you don't transmit the vibration into the inner ear, so you're not as likely to damage it from having the sounds being too loud. So those things, you should know what those are. And the other thing you should pay attention to on this, so let me clear at this point. Let's erase all the ink on the slide. The middle ear is sealed off here by the eardrum. So you have whatever air pressure out here. If you are going like driving up to Tahoe, the air pressure outside gets less and less. Or if you're diving underwater, the pressure out here gets more and more, which means that the pressure against the eardrum from the outside here can go up or down depending on what the air pressure is outside. And unless you balance that pressure from the other side, you are either going to implode your eardrum or explode your eardrum. Either case, it's bad. So we need to have a way to equalize the pressure on the two sides of the eardrum. And that's the job of this Eustachian tube, also called the auditory tube. It has a little opening. It's in the pharynx. Pharynx is just your throat. So there's a little opening on the inside of the throat, kind of up near the nasopharynx. And this Eustachian tube is often kind of flopped shut, but especially if you yawn or chew or swallow, it opens up and allows the air pressure to balance between the middle ear on this inside and the outside of the ear canal here. So, that's when your ears pop, is when you allow that Eustachian tube to open and let the pressures equalize. Right, if you hold your nose, the Valsalva maneuver, and blow, so we should if everybody hold your nose and blow gently, don't blow too bad, but you're going to feel your ears. I didn't see everybody hold their nose. Yeah, can you feel your ears, like your eardrums pooch out? That's because you're forcing air up this Eustachian tube. That's why if you are like on an airplane and you have like a bad cold and all of this is inflamed and it does not open well, you can have excruciating like pain because your ears can't equalize. People can actually get burst eardrums from having the inability to equalize if they're either going on an airplane or diving or something and they've got like some kind of cold. Should mention and that's when we talked about conduction deafness can happen if you have a burst eardrum from something like that. Conduction deafness can happen if you have an infection where this whole middle ear is filling up with fluid, right? Because if a bad cold or something, there's a little connection here, so all this mucus can actually get up into there as well. So that is a question.
Yeah, I just had a quick question in regards to what's happening with an ear infection and how that impacts this.
Ear infection can fill this with lots of goop, which makes it hard for this to vibrate and transfer the signal in there.
Sorry, go ahead.
And then there's more. Ear infections can be really dangerous because this is like kind of a really quick pathway to get up into the brain as well. And there's different versions of ear infections.
And Professor, what was you said the muscles, the stapedius and tensor tympanum tighten up stapes, and what was the other one?
The tensor tympanum basically grabs on the malleus and keeps the tympanic membrane from vibrating as much. And from its name, tensor tympanum kind of tenses up the tympanic membrane.
Okay, thank you.
This is kind of showing kind of schematically what's happening here. Sound waves coming in, vibrating the tympanic membrane, transferred through the little ossicles, these little ear bones, here through the oval windows, sending the vibration into the fluid of the inner ear, the cochlea. Here in the cochlear duct, this is where we're going to find the hair cells sitting on the cochlear, on the basilar membrane of the cochlear duct. That's going to actually be where the actual signal transduction happens. So we do still need to, we're going to need to talk more about that, but let's take a break right now. And when we come back, we will talk a little bit more about this, and then we'll get into our lab. All right, so we've been talking about hearing, and we've been talking about the outer ear, the middle ear. So now we're going to focus on the inner ear, the part that is inside the skull. So the inner ear actually is in a little cave in your temporal bone in your skull. Remember, that's how come bone conduction worked. Bone conduction worked because if you stuck a tuning fork on your skull and vibrated your skull, the inner ear is inside your skull bones in a little cave. And so that's why shaking the skull shakes the little hair cells in there and gives you bone conduction hearing. I can view image. Let's look at some pictures. So that this kind of gives you a sense, again, where we are. What we're actually talking about here is we have been looking at the outer and the middle ear, but now middle ear, outer ear. Now we're doing this little piece, and this is showing it in, much amplified, or much, very magnified. This thing that is kind of purplish here, that is what we call the Bony Labyrinth, the cave, the actual little cave that the ear lives in within your skull. So you imagine that's like the hollow space that's carved out of the inside of your skull bone. And then within that Bony Labyrinth, within that cave, is the Membranous Labyrinth. And here they make it kind of bluish. And those are the structures that you find inside the that cave, kind of like the soft body of a snail inside the snail shell. For the cochlea, you're going to actually have different ducts, different passageways that are both within the Membranous Labyrinth as well as in the space between the Membranous and Bony Labyrinth. So we're going to be talking about the tympanic duct and the vestibular duct and the cochlear duct. They're all just little canals that are either running through the Membranous Labyrinth or in the little spaces between the two. Some of the other stuff, when we get to the equilibrium senses, they're actually found just within the Membranous Labyrinth, like the Utricle and Saccule for detecting gravity are just within this area. This is called, they call the vestibule. In fact, let me just give you the three main parts of this, and then we'll go into each. First is the cochlea. That's the part we've been talking about that actually does hearing. Cochlea means, it's Latin for snail because it looks like a snail, kind of loops around 2.5 times. Obviously, it's got hair cells in there as well as other stuff that we'll pay attention to. And again, the vibration is going to come in the oval window via the ossicles, send the vibration up into the cochlea where it's going to get transduced into an electrical signal. And we have the little round window to equalize the pressure. So that will be hearing when we do the hearing. This area here where the utricle and the saccule, we call that the vestibule. I should get a different little font here. The vestibule. The reason why it's worth knowing that term is people will often talk about the vestibular senses. So like the sense of balance, we often call as vestibular senses, and they get their name because the detectors, the receptors that are detecting that stuff are here in the vestibule. Hanging off of the vestibule, kind of like little Mickey Mouse ears, are the semicircular canals, which we've talked about in lab a little bit, the things that detect the rotational acceleration of your head. Usually, when you talk about vestibular senses, you lump these in as well. So the utricle and the saccule are formally in the vestibule. The semicircular canals kind of emerge off of the vestibule, but we still, when we talk about vestibular senses, we usually mean the semicircular canals, as well as the saccule and the utricle, which are in the vestibule. So for right now, I'm going to put these on the shelf. I'm going to put these equilibrium or vestibular senses on the shelf for a few moments. And instead, we're going to spend a little bit of time looking at more detail. What happens when that vibration reaches the inner ear and gets up into the cochlea? And I will. So the basic way to think about it is you've got these ducts. There's this duct here. This is called the vestibular duct, and it loops around and around and around until you get to the very tip of the snail. The tip of the snail is called the helicotrema. When you get to there, then it spirals back out and then releases the pressure. So the pressure comes in, spirals up, and then spirals out on the other side. And as it's happening, the hair cells, which are inside this membranous labyrinth, are going to get vibrated, and they're going to send a signal to the brain. So what I'm going to do now is show a picture that shows the snail kind of unfolded, because it's kind of hard to see it when it's all like, you know, in a tight little loop here. So I'm going to here, this is the picture I was going for here. The basic idea on this picture is it's the cochlea, but instead of having all the other stuff, this is just the snail. And instead of having the snail in 2.5 turns, we have the snail in 1/2 turn, so you can actually see everything. Like I said, the vibration comes in one canal, goes up to the tip. This is called the helicotrema, the very tip of the snail. Helicotrema. And then the vibration comes down the other side and relieved at the round window. So this is the basic idea. Here in the cochlear duct, in this little place in between, we have the organ of Corti. This is where you actually have the hair cells. So these are the actual hair cells. And The last piece we need to add in here to make this a good story, the hair cells are sitting on a membrane that is actually important. So this thing I'm drawing in green here, this kind of boundary between this inner duct, the cochlear duct, and this tympanic duct where the vibration's coming back. That thing is called the basilar membrane. Maybe I'll draw it, do a different color. Do a hot pink. Wow. So this membrane here, so the hair cells, they're on top of the basilar membrane. I obviously can't draw very well with this thing. Wait, what's going on? Hold on. All right. The basilar membrane has some special properties that you need to know. Just like most things have some physical resonance frequency, like if I have a, in fact, here's a ukulele, and there are some strings that are a bit fatter and vibrate at a lower frequency, and there are other strings that are thinner and vibrate at a higher frequency. It's just their natural physical vibration frequency. Just it's due to their physical structure. If you just pluck them, there is a natural frequency they want to vibrate at. This one wants to vibrate higher. Boom. This one wants to vibrate lower. That's just the natural energy that it vibrates due to its actual physical properties. So we just call it like the resonance, the physical resonance of that particular structure. When you get to the basilar membrane, it has different physical resonances at different parts. When you're up at the helicotrema, this part vibrates at low frequencies, like oh. When you're up here, more near the oval window here, this is vibrating at high frequencies. Just due to the physical structure of this thing. The part near the helicotrema is kind of more floppy and it just tends to vibrate at a lower frequency naturally. The other part vibrates at a higher frequency. And it's not just about what frequency does it vibrate at when you excite it. It's also what frequency does it like to absorb energy at. So if I start like, if I start sing at this thing at the frequency that it is tuned to, it's going to absorb the energy from my voice and start vibrating sympathetically. But the strings that vibrate at different frequencies aren't going to do the sympathetic vibration. It's just the thing that's physically tuned to that frequency that is going to start absorbing the energy sympathetically when it is exposed to that vibration of the sound coming in at it. So it's the same thing in your ear. Like if you are hearing a low frequency, the basilar membrane up here, more towards the helicotrema, is going to absorb that energy and start shaking and jiggling. Which means as that happens, the hair cells that are on top of it in the organ of Corti are going to jiggle and move. And that's going to send a message to your brain that, oh, there's a low frequency. This part of the cochlea is vibrating. If you hear a high frequency, right, that is going to stimulate the basilar membrane more over here, more at the beginning of things. And then that's going to wiggle the hair cells on the organ of Corti up on this region of the cochlea. And that's going to tell the brain, oh, it's a higher pitch sound. So this physical tuning of the basilar membrane is kind of giving you the first.
Hello, Pablo.
Wait, hold on, we need somebody to mute yourself if you're.
I'm watching the dog.
Oh, it's muted now. Okay. So does that make sense? There is more going on than just this when you get into the more, you're going to get into more subtleties of how this works. And kind of breaks my heart. I'm not going to go into it. It's kind of actually the stuff that I did my dissertation work on as well, was looking at how the ear is a much more sophisticated filter than just these pure physical resonances. It actually, if all you had was this, you wouldn't be very good at detecting timing information and stuff. So there's actually a lot of other stuff going on in here. And in fact, not all the hair cells in the organ of Corti are actually receptors. Some of them are actually efferently controlled and actually change the properties of the system by actually being like little motors. So it's, the actual story is more complicated, but this is a good first approximation for what's happening here. You know, when people have cochlear implants, they rely on this, we call tonotopia, this kind of mapping of the frequencies, high frequencies when you first come in, low frequencies up at the very tip of the snail. If I have another, let me back. Whoa. Hold on, I gotta get my controls here. Like another one of these, like a cochlear implant. Basically, they have a little transmitter, well, actually a little microphone first, that is listening to what's going on in the room around you, and then sends it into this little thing, a little array of electrodes threaded up your cochlea, and it basically does a frequency analysis of the sound. And if there are higher frequencies, it's gonna do little electrical stimulations of the cochlea, whoops, down here, low frequencies up near the helicotrema, higher frequencies will be stimulating electrodes more here. And that's how a cochlear implant works. It just manually does a frequency analysis of the sound coming in and manually electrically stimulates the cochlea appropriately. And it's not going to give you perfect hearing of anything, but it will give you enough information that it'll make it a lot easier to navigate the auditory world. But that just gives you another idea of that tonotopy. High frequencies are shaking the basilar membrane near the beginning of the snail here, and therefore shaking the hair cells in the organ of Corti here. Low frequencies up near the helicotrema, near the apex of the snail. Again, there's so much more we can do about the ear, but we're not. I will mention one last thing just because it's really cool. I mentioned how not all the hair cells in the organ of Corti are just receiving. They're not just receptors. There's also efferently controlled ones that help do a much more sophisticated tuning of the ear. It turns out if you put a microphone in somebody's ear canal, you can actually hear sounds. They call them otoacoustic emissions. And it's because those little active hair cells are actually wiggling up and down, moving the fluid in here, which is then pushing and pulling on the ossicles, which then push and pulls on the eardrum. And the eardrum basically works like a little speaker cone and sends out these very faint but measurable sounds that are a result of that active activity inside your cochlea that you can actually record. So it's kind of trippy. So any questions about hearing? All right, so then the last thing we got to do are the equilibrium senses, and then we'll finally be done with the nervous system. So there's going to be the saccule and the utricle. Whoops. These basically tell you which way is gravity. We also call them static equilibrium. So static because even you're not moving, static means still. Even if I'm just sitting still, they're working and telling me which way is gravity pulling in relation to my head. We can also call them linear acceleration. Because linear acceleration is no different than gravity. Gravity is just you're accelerating down 9.8 meters per second. If I was in my Ferrari and I hit the gas and just go faster and faster and faster and faster, as long as I keep getting pegged against the back of my seat, these don't know the difference whether gravity's pulling backwards or whether I'm accelerating. So it's linear acceleration, gravity, it's the same difference. The other ones are the 3 semicircular canals. These tell you which way your head's spinning. So sometimes we call this dynamic equilibrium. So they only activate if you're moving, like, and there are three of them for the 3 basic, X, Y, and Z axes. Dynamic equilibrium, we also call them rotational acceleration. And I should mention the saccule and the utricle are basically the same thing, but just in 2 axes. Just like there's 3 semicircular canals. I'm not sure why they get different names, but in us, they basically are the same structure, but at 2 different axes. So you can tell gravity from different directions. And how do they work? They both work with hair cells. So let's go back to the, I'm gonna go back to the silverthorn. Let's see, slideshow. Here we go. I'm gonna go share screen. So let's start with the saccule and the utricle. The saccule and the utricle are basically hair cells with a bag of rocks on top. So saccule and the utricle and the saccule, they're sitting here in the vestibule. And what are they? There's the hair cells. They're in the spot they call the macula, like the saccular macula or the utricular macula. Macula just means the spot. It's like a little, imagine a little patch of grass, but instead of growing grass, you're growing little hair cell bundles. And then there's this membrane on top with a bunch of otoliths. Otoliths are little calcium carbonate crystals. So you can think of it as rocks. So think about hair cells with rocks sitting on top of their hair bundles. And that this little structure is anchored inside your skull, right? This whole dealy bopper is inside the temporal bone of your skull. So when you move your skull, this whole structure is gonna move with your skull. And here they're showing the dude, he's looking, head's neutral, gravity's pushing down, but now he's like, whoa, up in the sky, is it a bird? It's a plane. As he's looking up, all of a sudden the rocks are getting pulled down due to gravity because of the now angle of this thing, and the hair bundles are bending. And so the brain is getting information, oh, gravity is pulling that way. You know, if he looked down at his feet, the whole hair bundle would go now the other direction, they would bend the other way, and instead of depolarizing this way, they'd hyperpolarize because now they're going towards their away from their kinocilium. And so you're getting information about which way is gravity pulling based on the bending of the hair bundles due to the weight of the little rocks, the otoliths. Otolith just means like ear stones. Oto from ear, like litho from stone. So does that make sense? They're pretty straightforward. You know, in other animals, they do more. They can be, you know, there's all sorts of different things. So saccule and the bullfrog does some different stuff and does sensing vibrations and things. But for the humans, it's basically which way does gravity pull?
Dr. Egger, I had a question because my dad just had to go to the hospital for really bad vertigo because those rocks got loose. So do they just stay like that? Like, is it going to be like a problem now forever? Or
So that is a problem. In fact, that was something somebody did their research project on.
Yeah, I wanted to do mine on that.
So it's, I forget the name. There's actually a name for that condition. I'll look it up in a few moments. But basically, if for some reason these rocks get stuck and aren't moving properly, you're constantly feeling like things are messed up and things are pulling in the wrong direction or your orientation is off. There are some techniques that people use where they kind of move their head in one direction or another. There are different ways to try to loosen them up and get the system working again. So, but yeah, things can go wrong because they can get stuck or they can get knocked out of place from getting whacked in the head. So that's definitely a real condition, and I'll look up the name of it when we take a little break.
Okay, yeah, it was really severe. He had to call the paramedic because he couldn't even walk.
Yeah, and you can also end up getting really bad vertigo just from ear infections or like what we call labyrinthitis when we're going to see in this semicircular canal here, that's where you have the sense of turning your head. Like if you have an ear infection and things are kind of inflamed in here, you just always feel like you're spinning. You just always feel car sick. You want to barf just walking across the room. So the semicircular canals. Oh, where is it? So here are the semicircular canals. Again, the three of them are identical except for different orientations. Again, you know, they pitch, yaw, and roll. Basic idea, you have fluid in that tunnel, and again, we're not going to get into the details. We saw this basically in our lab already. You've got hair cells with this little cupula, this little gelatinous curtain here. And when you spin your head, the inertia of the fluid will push on the cupula from one direction or the other, bend the hair cells, and that tells your brain you're spinning in one direction or another. You know, so there's not much to it here. It's basically, it's, you know, again, we've gone over this. The inertia of this fluid in the canals makes it stay still, assuming that you're not just in a constant spin, spin, spin. And that inertia causes the cupula to get bent as you move your head. That bending causes hair cells to depolarize or hyperpolarize, sends a message to your brain saying, oh, my head's spinning. And depending on which semicircular canal it is, you know which axis you're spinning on. Depending on whether they're hyperpolarizing or depolarizing, you know which direction they're spinning. So that's the basic idea of these semicircular canals. And like I said, you can get these pretty debilitating conditions. They call it labyrinthitis because we talked about the membranous labyrinth. So if there is swelling in here, sometimes it's secondary to like an ear infection. Like I had it once secondary to an ear infection, and just, yeah, I can say for a couple of days, I was dizzy all the time and had to try not to barf in class. Some people get it and it's not clear what it's from. It just comes and they can try to control it, but it's pretty debilitating. They just always feel car sick all the time and they don't know how to make it stop. You know, I say car sick. It's kind of that, you know, people have experienced that where you have this kind of mismatch between what you're seeing and what your head is telling you, and you start getting all nauseous and feeling bad. So I think I'm going to leave that there. Equilibrium senses, the dynamic equilibrium would be these guys, these semicircular canals. The static equilibrium, the utricle and the saccule, telling you which way is gravity pulling. Again, we often just call them the vestibular senses as well. You know, these also, and all your reflexes start getting worse as you get older. A lot of times when people are like elderly people are more likely to fall over because their reflexes involving these vestibular senses aren't working as well or as quick, and they're more likely to lose their balance. You know, a lot of times, you know, as people get older, they do more little exercises to kind of work on these and keep them sharp. So that's that. You should definitely know the idea of how is hearing transduced, right? It starts as an air vibration. The stimulus is the air vibration coming in. Then hits the tympanic membrane. The eardrum vibrates the eardrum. The eardrum's kind of like the first place of the energy getting into your system. Then that vibration is transferred by the ossicles into the oval window, so it gets into the inner ear. Now, once we're in the inner ear, now that vibration is going up these ducts within the inner ear. We have, again, I didn't really mention the vestibular duct, down to the helicotrema, out the tympanic duct. The main thing to realize, though, is that the basilar membrane is going to be maximally excited at a certain frequency, whether it's a low frequency, which is going to be down near the helicotrema, a higher frequency closer to the beginning here. As it wiggles and jiggles, as it absorbs that vibrational energy, the hair cells that are sitting on top of it, again, the hair cells here are sitting on top of the basilar membrane. As the basilar membrane jiggles, then the whole hair cells jiggle here, and that's going to cause an electrical signal going to the brain, telling the brain there's a vibration coming in the ear and the frequency range is such and such, depending on, you know, whether it was down here or down here. So are there any questions about that? So again, as we go on in the semester and there's a little more time, maybe I'll talk a little more about there's lots more we can talk about this, but this is kind of the core of it. All righty. So I can say, let's see. So nervous system, CNS, check, PNS, afferent, check. All right, we did the efferent as well. All right, so which means check. Obviously, we're going to be coming back to the nervous system. The nervous system is playing key roles in keeping your blood pressure going. We're going to see all sorts of neural control of blood pressure in the next week. The nervous system is in control of operating your muscles, which we're about to talk about in the next class. The nervous system is constantly doing stuff. The nervous system is going to be at the key core of the endocrine system. We talked about the hypothalamus controlling the pituitary, which controls the endocrine system. So in fact, again, like I said, people often say like the neuroendocrine system because they're so inextricably entwined with each other. So just because we're leaving the nervous system behind doesn't mean this is the last we've seen of the nervous system. All righty.